In this video, we will have a broad overview of the structure and function of the nervous system. The primary functions of the nervous system are to detect information, analyze information, and to transmit information throughout the body. There are two major cell types in the nervous system: the neurons, which are the functional cells of the nervous system, and the neuroglia, that provide protection and metabolic support. The nervous system is divided into two major components. The central nervous system is made of the brain and the spinal cord. And the peripheral nervous system is made of the cranial nerves and the spinal nerves. Neurons are the functioning cells of the nervous system. There are two major types of neurons. The first are the afferent neurons, also called the sensory neurons. The afferent neurons transmit information to the central nervous system. The efferent neurons, also called motor neurons, carry information away from the central nervous system. While these two types of cells look a bit different, you can see that they share the three major components of a neuron. The first is the cell body, also known as the soma. This contains a nucleus with one or more distinct nucleoli and a well-developed endoplasmic reticulum. Around the cell body, you also see dendrites. These are branched extensions of the nerve body that will conduct information toward the cell body. And the dendrites are the main source of information for the neuron. The dendrites and the cell body have a lot of synaptic terminals to allow communication with other neurons. And then we have the axon, which you can see here are the long efferent processes that project from the cell body. Most neurons only have one axon, but they may have multiple branches that could lead to multiple axonal terminals. The axon is where the nerve impulse goes from the cell body to the synaptic terminal. In the central nervous system, the neuroglial cells provide protection and metabolic support. The most numerous of these cells are the astrocytes. Astrocytes provide regulation, and they often are found between cells to help provide communication and other support for the neurons. In addition, you can see here where this astrocyte is an important part of the blood-brain barrier. The oligodendrocytes help protect the nerves by providing that myelin sheath that wraps around the axons of the nerves in the central nervous system. Our central nervous system also has a resident immune cell called the microglia. These cells have phagocytic properties much like a macrophage. And then finally, we have the ependymal cells. These cells you can see here on the left of this picture line the ventricles that houses the cerebrospinal fluid. In the peripheral nervous system, we have two types of neuroglial cells. The satellite cells provide a basement membrane so that large molecules are not able to diffuse into the nerve. And the Schwann cells provide the myelination in the peripheral nervous system. You can see here the myelin is built in segments with small gaps in between. These gaps are called the nodes of Ranvier, and this is where you find concentrations of voltage-gated sodium channels to help propagate the nerve impulse that travels along the axon. Our brains are needy, and they require constant attention. Our brains receive 15 to 20% of the resting cardiac output, or about 750 milliliters of blood per minute. It also consumes 20% of the body's oxygen. And because our neurons cannot store glucose, these cells have a constant need for delivery of blood glucose to the brain. At the end of our nerves, they end in a synapse, and this is a structure where the neurons communicate. There's two major types of synapses. You can have an electrical synapse, and here there are ions that travel through the gap junctions, or the space between the synapse. In addition, you can have a chemical synapse, where here you have this synaptic cleft, and the neurotransmitters will diffuse from one nerve to the other. These neurotransmitters can have an excitatory response, or the binding of the neurotransmitter could lead to depolarization of that nerve membrane. Or it could be an inhibitory response, where binding of this neurotransmitter to a receptor leads to hyperpolarization of that nerve membrane, so you're not able to generate a new action potential. In the neuron, neurotransmitters are synthesized in presynaptic neurons, and they're stored in the synaptic vessels. Presynaptic neurons are just neurons that lead into that synapse. The activation of a nerve impulse will stimulate that presynaptic neuron, and this leads to movement of the synaptic vesicles to the cell membrane. Ultimately, this will lead to neurotransmitter release into the synaptic cleft. Once in the synaptic cleft, this neurotransmitter will diffuse across that cleft towards the postsynaptic neuron, and it will bind to a postsynaptic receptor. It's important to have control of this synaptic function, and so this will require the removal of the neurotransmitter from the synapse. This way you don't have excessive or inappropriate neuronal impulses being made. To remove the neurotransmitter from the synapse, there's three things that can happen. One, we can have reuptake. This is where the neurotransmitter is taken back up into that presynaptic neuron. The second, it could just diffuse out of the synaptic cleft and no longer be available in that space to bind to the receptor. Or three, it could be broken down by enzymes that will target that neurotransmitter. These three mechanisms are important ways to make sure we can turn off these neuronal signals when needed. And this is our brief overview of the structure and function of the nervous system.